Good afternoon, everyone. My name is Anvita Gupta, and today I will be presenting on a novel method of targeting intrinsically disordered proteins for drug discovery with applications to cancer and tuberculosis. For many years, the traditional view of proteins was the sequence to structure to function paradigm, which states that a protein's amino acid sequence codes for a stable tertiary structure necessary for the protein to function properly within the cell. However, in the last decade, a new view of proteins has emerged with the discovery of intrinsically disordered proteins, or IDPs. These proteins change shape constantly with no equilibrium values, as you can see on the protein on the right. These proteins play a key role in disease. 70% of all cancer proteins are disordered, and there were 1.6 million new cases of cancer in 2013 alone. Key proteins in Alzheimer's, tau, and beta amyloid are disordered, and proteins linked with tuberculosis, like CAT-G, which you can see in this photo here, are also disordered. 5 out of 7 proteins in Ebola are disordered, like VP40 over here, and this makes IDPs extremely attractive drug targets for drug development. Because they change shape constantly, IDPs are able to bind to many different proteins within the cell, as you can see in this graph of the protein-protein interaction network of PTEN. PTEN interacts with several different proteins involved in cancer, and its mutation affects all the different proteins in its network and leads the cell towards disease. But though IDPs are attractive targets for drug development, they're also extremely difficult to target with drugs because they change shape constantly. How do you find a key that fits into a constantly changing lock? We propose to inhibit IDPs in a different manner. IDPs bind to other proteins through hair-like projections called morphs, or molecular recognition features. These morphs become ordered when the IDP binds to another protein. We propose to inhibit the interactions of the disordered protein. How do we do this? By finding a drug that mimics the disordered protein's morph. The drug can bind to the IDP morph's binding site instead of the morph and block the IDP from binding there. So our objective in this project is to develop a method to target intrinsically disordered proteins for drug discovery, apply and evaluate its effectiveness on diseases in which IDPs are mutated. The approach we will use is interdisciplinary. As I mentioned earlier, we find drugs that mimic the IDP morph to inhibit the IDP's interactions, but also we integrate biomimicry with machine learning to create a classifier that can predict drug activity against disordered proteins. And furthermore, we validate this classifier and apply it to find new drugs for diseases in which IDPs are mutated. Here we get into the data set we use for our project. We use 1,700 drugs to train our machine learning algorithms from 4 bioassays conducted against 3 proteins shown above. Approximately 10,000 approved and experimental drugs are used for virtual screening to find new tuberculosis drugs. In addition, we use molecular docking to simulate the binding of the drug to the protein, and we use R and Python to calculate Euclidean distances and code our machine learning algorithm. The specific algorithm we use for biomimicry is the support vector machine, which draws a decision boundary between positive and negative data points, as you can see here. We train our classifier with a 50-50 ratio of actives to inactives, and we separate 20% of the data into an independent test set, and moreover, we code our algorithm 100 times. So I say that the drug will be active if it's similar to the IDP morph. How do we quantify the similarity? The answer is the spectrophore. The spectrophore consists of 48 3D features describing atomic partial charges, atomic electrophilicity, atomic lipophilicity, and atomic shape deviations. We compute 4 Euclidean distances between the spectrophore for the drug and the morph, and these are used to denote similarity. Here are the results of our classifier. We had an accuracy of 91% on the training set, 90.5% on the test set, and the area, which shows that our algorithm generalizes well to new data, since those two numbers aren't very different. The area under the ROC curve provides an alternate measure of accuracy, and an ROC curve for one of the SVMs is shown here. This particular SVM had an accurate area of 0.946. For all the SVMs, the area under the ROC curve for the training was 91.2%, and for the test set, it was 0.907. The sensitivity was 88%, and the specificity was 93%. And this picture on the right shows how our classifier is able to accurately discriminate between actives and inactives. Active data points are graphed as circles, inactive data points are graphed as triangles, and the predictions made by our classifier are shown as colors. Now that we've created our algorithm, we want to validate our method by applying it to the cancer protein thrombin. Thrombin promotes tumor metastasis, and its most potent natural inhibitor is the disordered protein hirudin. So we screened 3,000 FDA-approved drugs by their similarity to hirudin, and we rank them by their probability of being active. The top drug out of all of these 3,000 was dabigatran, and dabigatran is an FDA-approved thrombin inhibitor, and this shows the validity of our algorithm. On the right, you can see dabigatran bound to thrombin. And now that we've validated our algorithm this way, we want to apply it to find new drugs for tuberculosis. Tuberculosis affected 9 million people in 2013 alone, and is estimated to lie dormant in 1/3 of the world's population. So now I'll ask you to look to your left, look to your right, and look at yourself. Chances are one of you has it. Specifically, we target tuberculosis by targeting the protein EMBER. EMBER has an FHA domain, which is important for prokaryotic signaling. And moreover, as you can see in the diagram on the left, it regulates production of the proteins EMBB and EMBA, which are important in arabinogalactan production in the bacterial cell wall. FDA-approved drugs like ethambutol target EMBB and block this pathway, but EMBB mutation causes drug resistance. And that's why we target EMBER, because that's a viable way to overcome this problem. In addition, we screen our 10,000 drugs against tuberculosis protein 39.8, which also has an FHA domain, to eliminate false positives. And we screen our drugs against PTPA from Staphylococcus aureus to find broad-range antibiotics. First of all, we validated our method on FDA-approved tuberculosis drugs. As you see here, ethambutol ranks the lowest, and this makes sense because if ethambutol targeted EMBER, we wouldn't have the problem of drug resistance. In addition, our approach was able to filter out decoys like tigecycline. Tigecycline targets many members of the Mycobacteria family, but not Mycobacteria tuberculosis. Because tigecycline ranked as only 0.22 against TB 39.8, we were able to filter it out. Most importantly, though, we found 12 top new tuberculosis drugs that can be used to target EMBER, and which are shown here. Here you see the prior experimental evidence of these inhibitors and the details of which bacteria they target. And several of these compounds are antibacterial, and they ranked highly on all 3 of the proteins that we tested them against. Moreover, we integrated our predictions with prior experimental evidence for ranking and for validation. So this heat map shows the prior experimental evidence we integrated with. The first is the number of interactions each drug has with proteins related to EMBER. The second is the number of times each drug is listed as an inhibitor in the literature. And the third is the absolute value of the docking score of each drug with EMBER. And this heat map adds the evidence of prior inhibitory activity and lets us distinguish between our top 12 drugs. So our top ranked inhibitor, zinc 9600632, which I will call zinc 032, also had the highest docking score. 2 of the drugs, zinc 683 and zinc 083, have prior experimental evidence against tuberculosis already. Zinc 083 was experimentally validated as an inhibitor of TB thymidylate kinase. In addition, 6 of these drugs have prior experimental evidence against actinobacteria, the family to which tuberculosis belongs. Our SVM has high accuracy and generalizes well to new data based on only 4 Euclidean distances between the drug and the morph. Here you can see the top-ranked drug, zinc 032, bound to EMBER, its protein receptor, shown as a mesh. So in conclusion, we combine biomimicry and machine learning to predict drug leads to target IDP interactions. We can use this method, as we've done here, to reposition existing drugs, which cuts down the failure rate in clinical trials, reducing the time and cost of drug discovery. Since several proteins of Ebola are disordered, we are currently working on extending this method to find potential antivirals to combat the recent Ebola outbreak. And this method can also be used to find new drugs to be synthesized. The tuberculosis drugs we found are going through preclinical trials with their collaborators in China. So here I show one of our key findings. which is that while an IDP morph binds to an ordered protein and leads to disease, by finding a drug that mimics this IDP morph, we can bind to an ordered protein and instead lead to treatment. So we show that IDPs can be inhibited by targeting their interactions with other proteins, and that drug activity can be correlated with a small number of features describing the 3D field properties of a molecule. We validated our method of drug discovery by applying it to the cancer protein thrombin, and we are working on applying our method to find new antivirals to combat Ebola. Most importantly, we found 12 new drugs for tuberculosis by screening and ranking approximately 10,000 FDA-approved and experimental drugs. And we ranked these drugs by prior experimental evidence of their activity as inhibitors. These 12 drugs are so promising that they've been moved to preclinical trials with our collaborators at the Chinese National Academy of Sciences, and a paper on this research has been submitted to Nature Genetics.
1 minute.
Here are my references. I'd like to acknowledge my mentor, Professor Gil Alterovitz of Harvard Medical School and MIT. I'd like to acknowledge Harvard and MIT for providing a platform for my research. I would like to thank Dr. Sangeeta Agarwal at Wright State University for guiding me in this area for many years. I'd like to thank Basis Scottsdale, my high school, Siemens and Discovery Education for putting this event together, and the George Washington University for being gracious hosts. Finally, I'd like to thank you all for your time. Thank you.